All right, welcome back to our review on macromolecules. Here we're just going to put it all into one table. It's going to be very succinct, so you're not going to get all the detail that you have on your notes, but it's going to give you a good overview and a global view of what these macromolecules are and where they play and how they function and what they're made of and who has what monomer and all that good stuff. So let's start writing out this chart. If you already have it, hopefully print it out. You can fill it in as we go along, or if you've already filled it in, you can make sure you got it all right. Either way, it totally works. So I've done my best to recreate this on the board. So the first macromolecule we learned about, we're going to go in order of how we learned, is carbohydrates. So the first one is carbohydrates. And the monomer for carbohydrates is, the best monosaccharide we know is glucose. The linkage between glucose monomers, remember, was a 1,4 glycosidic linkage. Now remember, there were 4 different types of macromolecules, and the types are going to help lead to the function, so we're just going to carry it forward. So we had starch, we had glycogen, we had cellulose, and we have chitin. So if you carry these forward over here, the function of starch, that was our storage polysaccharide for plants. So plant storage polysaccharide. I'm going to put plant storage poly over there. And you had the alpha helix shape, just so you can remember what the shape was. You have glycogen. This was your animal cell storage polysaccharide. And this was the branched alpha helix. Do that so you can remember. Cellulose, this is our insoluble fiber that we can't digest, but that's okay, it's still good for you. But it happens to be the plant structural polysaccharide. And remember, cellulose was a little bit different. It wasn't made of alpha glucose, but it was made of beta glucose, which gives it a totally different shape, a totally different structure, different function. And in this case, instead of being an alpha helix, it was kind of the chains, and they get a whole bunch of those together, and you get your microfibril structure inside the plant cell wall. And then lastly, chitin. This is your animal structure, specifically your arthropods, their good old exoskeleton. All right, just quick bullet points, and what you'll be required to know is a lot of detail underneath all of these, but this is just to give you that big overview picture of what you need. All right, so that is macromolecule number 1. Now we're off to number 2, the lipids. Now lipids are a little bit different. Remember, they don't have, they don't make polymers, but they basically aggregate together. So when it comes to monomers under lipids, we can't really say that there's one monomer for a lipid. So let's just basically write out our train of thought here, and we'll just say does not form polymers. Rather aggregates together due to hydrophobicity. That's a decent enough description, I would say. Now, remember, when it comes to linkages in lipids, we do have one lipid we can talk about, or actually 2 lipids that we can talk about. We do know the linkages between them. When it comes to fats and phospholipids, there are ester linkages within itself. So you have ester linkages, and now when it comes to different types of fatty acids, or to different types of lipids. Now we have our fats, which are the triglycerides. And underneath fats, you have saturated or unsaturated. You also have phospholipids. And lastly, you have steroids. Now let's talk about the function of each one of these. When it comes to fats, the function of fats is for energy, protection, insulation. Those are your big things. So you have energy, you get a lot of energy out of fats. You actually get more out of that than carbohydrates for sure. Energy, insulation, and protection. Sometimes it's good to have a little bit of protection. And then phospholipids, this is the component of the cell membrane. And then lastly, steroids, that would be cellular activation. That's a broad definition for steroids, but it works. Cellular activation. Okay, so those are the first two. I need to erase this to make room for the last two. But we have carbohydrates, we have lipids. These are the basic things. So now you can kind of group together who's with who. Oftentimes I'll see students swap things in and out, but try to keep them together. Okay, that was carbohydrates and lipids. Next, we got proteins and nucleic acids. Okay, number 3, proteins. The monomer for proteins. You only have one monomer in proteins, and that is your amino acid. The linkage between amino acids, remember what that was? It was a peptide bond. Now I didn't go into the different types of proteins out there, but what you do need to know is that the types and the functions kind of go hand in hand for proteins. So at this part of the table, it's going to look a little bit different. But I will give you a little bit of information on the types of proteins. The major types of proteins you have enzymes, and you have structural. Those are the two that I really want to focus on. There are others out there, but, and actually, we'll do transport as well. Because we didn't talk about these because we stopped here after we talked about a polypeptide chain folding up to become a specific shape so it can be a certain kind of protein. There are thousands of different kinds of proteins in your cells. So these are like the 3 main types of proteins you'll see in a cell. Enzymes, their function is to basically be protein catalysts, if you will. They will enable reactions to happen inside your cell. So these essentially are catalysts for cellular reactions. A lot of stuff in our cells can't do what they need to do unless you have a protein present. And so like the dehydration reactions we talked about, the hydrolysis reactions we talked about, all of those reactions are happening because proteins, enzymes, which are proteins, are grabbing a monomer here and a monomer here, and they're putting them together. Those are proteins that are doing that, and they are catalyzing the reaction to happen. Another type of protein would be structural, and these basically maintain cell shape. And then transport, these proteins literally transport other molecules around the cell. So transports molecules. So there's lots of things going on inside the cell. Proteins do a lot of work. Without functioning proteins, our cells can't work at all. So that's proteins. You don't necessarily need to know about these functions yet, but we will be talking about them in different aspects of different parts of biology as time goes on. So it's good to get a good introduction right now. And then lastly, nucleic acids. The monomer for a nucleic acid is your nucleotide. Remember, there's a couple linkages I want you to know about. The first one is a phosphodiester bond. That's between nucleotides, and hydrogen bonds between bases. So there's a couple things going on. When it comes to the types of amino acids, that's easy. You only got two. You have DNA and RNA. So DNA, RNA. And the function of your DNA, this is your genetic material. Essentially codes for all your proteins that are happening, that are doing their jobs inside your cell. RNA has multiple functions. We didn't realize how many functions it had until, I don't know, the last 20 years. It's pretty amazing. So, but the main function, it's the go-between between your DNA and your protein. So we're going to just allow it to have multiple functions right now, but what I do want you to know is that the main function that we're going to study is it helps code the protein, code for protein. That's a very broad, and I'm not too happy with that definition, but it does the job for now. You don't need to know this for the test, but knowing that your nucleic acids ultimately serve to create protein, that's what's going on. All right, so that is your overall view of these last 4 macromolecules. So I'm hoping that helps put in perspective. Here's your macromolecule, here's your monomer, here's the linkage between them, and here are the different types that you can have within them. So you could have them all on one sheet, and then you could go back into your notes and study them in more detail. So we'll be having our test, and good luck, and I hope to see you for the next lesson. We're going to be talking about water and the lovely properties of water that are coming up next. So thank you again. Good luck on that test. See you soon.